So first, I would like to thank the Andrew Workshop Committee to give me today's opportunity to present my work. So, work about the culture of a new enteric neuron culture to study the effect of EVs, the extracellular vesicle, in autism spectrum disorder. So, as you know, ASD, it's a neurodevelopmental disease with behavioral symptoms, with altered social interaction, communication, and the causes are multifactorial with genetic and also environmental factors. And it's associated with abnormal neuronal connectivity and activity in brain. But it's also correlated with gastrointestinal symptoms. In fact, around 70% of ASD children develop GI symptoms, constipation, diarrhea, pain also. And more interestingly, there is a correlation between the severity of behavioral symptoms and GI symptoms. So we know that in brain, so we know that in brain there is an alteration of synaptic connectivity, and we are interested to see if by a mirror effect there is the same thing in the ENS, the enteric nervous system. So the ENS, it's the nervous system that innervates and also regulates the gut function. And we know that there is an alteration in intestinal microbiota composition in ASD patients. And in the laboratory, we found that the transfer of microbiota from ASD patient to mice induced GI and behavioral symptoms. So if you would like more information about this, you can see the Justine Marchic's poster. So, but we don't know exactly what is the mechanism and how the microbiota can communicate with ENS in ASD. So, we propose to study the microbiota-derived extracellular vesicles. So, the EVs are produced by both eukaryotic and prokaryotic cells. They participate in intercellular and inter-organ communication, and they carry a lot of molecules like protein, nucleic acid, and also lipids, and they are a signaling cargo. Also, recent publications show a link between the profile of eukaryotic EVs and several neurological pathology. But we don't know if the microbiota EVs are implicated in ASD, and also if they can communicate with the enteric neuron. So the hypothesis here is that the microbiota-derived extracellular vesicle play a role in enteric neuronal connectivity and activity in autism spectrum disorder. So for this, we first develop a new model of enteric neuron. Like you can see here, we have a neuron. We have a neuron culture with a very high density and a very dense network. And please, if you can play the video here. No. Okay. So you can see also that these neurons have a very strong spontaneous activity. We have a cohort of patients with adult control and ASD, and we isolated the EVs by several steps with notably centrifugation and ultrasonography, and we characterize these EVs in this cohort of patients, and by electron microscopy, Western blot, and also nano-tracking analysis. And like you can see here in the graph, we see a different profile between control and ASD patients on the size of these EVs, and also in the composition of short-chain fatty acid with acetate. Okay. Okay. And then we apply this EVs on the culture of enteric neuron to see if they can modify the activity of this neuron. And you can see here in calcium imaging that the EVs induce an higher and longer activity of enteric neuron. And after 48 hours of treatment, we saw the downregulation of the PSD99, a protein implicated in the synaptic connectivity. So in conclusion, in this study, we characterize the feces derivative EVs in control and in ASD patient, and we saw a different profile between these two groups. We developed a new model of enteric neuron culture to test these EVs on this neuron, and we saw a change in neuronal activity and a change in neuronal activity and the expression of synaptic protein. Thank you. Thank you for your attention, and thank you for all my colleagues and collaborators. Thank you.
Bravo.
Merci.
Pour avoir réussi. Merci. Est-ce qu'il y a des questions dans la salle sur ce topo court?
Merci beaucoup pour cette présentation. Qu'est-ce que tu penses de certaines études récentes qui disent que oui, les personnes autistes, en fait, ça ne sert à rien de travailler sur leur microbiote parce que, en fait, ça serait, les variations de microbiote seraient liées à des préférences alimentaires. Et donc, c'est une question un petit peu iconoclaste que je vais te poser, mais c'est justement, enfin, vu la qualité de ta présentation, je pense que tu pourras défendre la communauté. Voilà, pourquoi est-ce que, malgré ces résultats récents qui ont eu beaucoup de retentissement, c'est utile selon toi de poursuivre ces recherches?
Alors, je pense que c'est vraiment utile, d'une part, parce qu'on sait que dans d'autres pathologies qui ne sont pas du tout neurologiques, telles que les Mickey, il y a une très forte comorbidité et donc des douleurs chez les patients, mais également des problèmes de dépression, et ça affecte énormément leur qualité de vie. Et donc, je pense que travailler sur le microbiote des enfants autistes, notamment, ça pourrait vraiment les aider à aller mieux. Ça ne va sûrement pas guérir complètement la maladie, mais je pense que c'est vraiment important d'améliorer leur qualité de vie. Et je pense que là-dedans, ces études peuvent vraiment s'intégrer et vraiment améliorer la qualité de vie de ces enfants et de ces patients.
Et puis, ce que j'ai beaucoup apprécié dans ta présentation, c'est que au-delà de l'association variation de microbiote, de microbiome et puis trouble du neurodéveloppement, l'équipe cherche à montrer un lien mécanistique entre les deux. Et voilà, bravo à toi, merci beaucoup.
Merci beaucoup.